Hi, I'm Christophe Poulet. I'm a molecular and computational biologist at Liège in Belgium, and I'm here to talk about proteomics.
I'm Peter Katsikis. I'm a professor in immunology at Erasmus University Medical Center, and I'm involved in the research that involves proteomics in the ImmunAID project. So as part of the ImmunAID project, one of the questions that we are asking is to understand, to perform proteomics, which is trying to understand the expression of proteins in the plasma of patients, so in the blood of patients. Now proteins are the building blocks of cells, one of the building blocks. So by being able to analyze in the plasma the composition of what proteins are present, we can try to understand what's happening in different parts of the body. So you can imagine that in the situation of autoinflammatory diseases, when there's inflammation in different parts of the body, different cells or different tissues or different organs, they will be releasing different proteins in the blood. And being able to analyze in the blood what type of proteins are circulating in the blood, we can try to understand potentially what is happening at the level of the disease, the autoinflammatory syndrome. We can also start to understand maybe which tissues are being damaged and maybe also the mechanism behind this damage. So proteomics is really a tool where we do a very general and broad survey where we try to analyze as much protein as we can in the blood and find how many of these building blocks are circulating. So as part of the proteomics analysis that we're doing, one approach is to try and measure those building blocks, the proteins, with a technique which doesn't know anything beforehand of what's in the blood. So that way, Christophe is basically going to be breaking up proteins and trying to find out what they are.
Yeah, so basically with all those building blocks, we're going to isolate them and find a way to shatter them into pieces. And those pieces, we call them peptides. And those peptides, we're going to send them through a mass spectrometer, which will basically measure the weight of each peptide and also the charge. With all the charges and the weights, we can find out which peptides we are in presence in the blood. And after that, we recompile everything and find out what type of proteins there were in the sample.
The second approach that we're taking, which my group is taking, is we actually use an assay that looks for specific proteins. And this assay is one of the cutting-edge technology. It looks for 5,000 proteins, and it tries to find those 5,000 proteins in 60 microliters of blood. You only need 60 microliters. And in that tiny amount of blood, we can measure those 5,000 proteins. Are they up? Are they down? Now, what we can't find is things that are not on the test, what we're looking for. So these two techniques that we have with Christophe are complementary. Christophe looks to discover maybe the same proteins we're looking at, but also proteins which are not in the test that we're looking for. So he can find unknown things and new things. We're looking for very specific things, which are 5,000. So this is really cutting-edge technology combination that we're doing to find out what is happening with the proteins in situations like autoinflammatory syndromes.
Obviously, we're looking for the levels of production of proteins in the blood of patients. And so proteins can be very highly produced or lowerly produced. And so those differences, we are looking for them. And we will be happy to see if proteins are very differentially expressed or produced between categories of samples. If we can gather sample and classify sample and those patients in two subcategories, then we can go ahead and get some insight about what we should seek for the pathology, what will be the next treatment, what will be the target of future therapies.
So what Christophe just mentioned is, looking at the proteome, we can get a lot of information about patients with autoinflammatory syndromes. And as I mentioned also earlier, by looking at the proteins, we can maybe understand what tissue is being damaged, what's the mechanism behind the damage. So the goal, and what we would be really happy if the proteomics gives us the results we want to get for the patients, is potentially we could come up with new diagnostic tests. Maybe we can be able to classify the patients better. Maybe we understand what is the mechanism behind the disease, where we can come up with better treatments if we do understand that. maybe we can get diagnostic markers where we can predict who's going to get sick, or where we can detect a flare of the disease very early, so we can intervene and hit the disease hard and fast, so we get the best benefit for the patient. So there's many multiple potential diagnostics and therapeutic benefits by looking at the proteome, and that's what ImmunAID wants to achieve.